This is your lecture on protein synthesis. Protein synthesis is broken down into two parts, transcription and translation. The first part, transcription, is making an mRNA copy of the DNA in the nucleus of the cell. This is done by creating a complementary strand based off of the DNA using uracil rather than thymine. So this is how all RNA molecules are made, mRNA, tRNA, and rRNA. And during this particular process of transcription, we're going to talk about DNA being copied into mRNA because that's what is used to make the proteins. The enzyme that does that is RNA polymerase. It unzips the DNA, and then the RNA is copied from a complementary standpoint, and the DNA then zips back up. So here you can see that the adenine is pairing with uracil. Since this is RNA, uracil is used as opposed to thymine. Cytosine and guanine still pair up, and had this continued on in the illustration, these would be adenine here pairing up with the thymine. Before we move on from this is really the simplest part. We're going to go ahead and watch a video on transcription. So here's the RNA polymerase. This promoter sequence is where it tells it where to start, and this would be your DNA. The RNA polymerase opens up the DNA, and a complementary strand of mRNA is produced, and the DNA zips right back up, and now you have your mRNA strand. The terminator is what tells that RNA polymerase where to stop, and the process is complete. It's fairly simple for this part of the process. After transcription comes translation, and this is where the actual protein is made. It's a little bit more complicated. The mRNA leaves the nucleus and goes to the ribosome, and the ribosome uses that code to pair up tRNA with the amino acids that string together to make the protein. So mRNA decodes the nucleotides into a sequence of amino acids. That's kind of the definition for translation. The sequence is taken to the ribosomes and build the proteins. The ribosomes are what read the codons, that's the mRNA, and decode it into the tRNA. And the tRNA goes and fetches the correct amino acid, and then those amino acids are chained together to make the protein. Here's a picture of what's going on. This part is the transcription, where the mRNA is made and then it leaves the nucleus and goes to the ribosome, which is this blue blob right here. In the ribosome, the tRNAs, which are these little T-shaped guys here, pair up complementary with the mRNA. At the end of each tRNA is an amino acid. A new one will come in that pairs up here and drop off its amino acid, and then it leaves. And then another one come in, will drop off the amino acid and leaves, and then we'll continue until we run out of mRNA and reach the stop codon. Then we'll have this long chain of amino acids, which is a protein. Here is a video showing that process.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into three stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found in the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically A and G. Attached to the end of the tRNA is the corresponding amino acid, with methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
So there were more details in that video than you really needed to know. Things about like the P site and the A site. Those are more detailed, but you got the idea and kind of saw a visual of what that process looks like. So now that you've watched that, try and look at this picture here and describe to somebody else around you, a family member, someone, what is happening in this picture. Pause the video to do this because it will really help you to better understand this process. Before we move on to the genetic code, I'm going to show you a video of the real-time process, how fast this actually happens in the cells. So this video is a little longer, very interesting.
The central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it sneaks away from the nucleus into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific three-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in, RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second.
All right, so that gives you another visualization of how fast that process actually works. So within the genetic code, the letters are grouped together, or the nucleotides are grouped together in groups of 3. On the mRNA, that group of 3 is called a codon. Each codon codes for an amino acid, and there are 20 amino acids that are possible to be found in proteins. Some of them we make in our bodies, and some of them we eat. So this long string of letters here is like a genetic message without punctuation or spaces between the words. So we can break it down into groups of 3, and each group of 3 can represent a protein, or an amino acid that combine together to make a protein. There's also start and stop codons that you saw in those videos. The start codon is methionine. It's always AUG. So each protein is going to start with methionine as its first amino acid, and the code that codes for it is AUG. The stop codons vary. There's 3 of them, and these do not have an amino acid that is associated with them. So it's just the stop. It's like a period at the end of a sentence, not coding for any of the amino acids at all. So mRNA is codons, and tRNA is anticodons. These are complementary to one another, and we know exactly which codons code for which amino acids. So for example here, if we were looking for the protein, or the histidine, which is CAU, we could first find the first base on this side is C, second base up here is A, so then we go to this box where C and A meet up. Third base over here is U, histidine. Or if we were looking for GAC, first base on this side is G, A up here, so in this box C is aspartic acid. You don't need to memorize this table by any means, but you should be able to use it. So make sure that you understand how to find an amino acid based on the sequence, but also what sequence based on the amino acid. So you should be able to go in both directions. Remember that this table is mRNA. So if you are given DNA, you first have to translate it, transcribe it before you're able to translate it into these amino acids. Let's practice that. So here's a DNA sequence. You're going to first transcribe it, turn it into mRNA, and then translate it into the amino acids using that table. You might want to pause the video. That table is also available in your textbook, but please use this for practice before you look at the answers. You might be surprised at how effective that can be in helping you to understand the concept. Pause the video now and try and do these 3 tasks. Now that you've done that, check your work. So the complementary mRNA codons for this tRNA strand are as such: AUG, CCG. Notice that there's U's instead of T's. That's because this is RNA rather than DNA. This would be the complementary tRNA codons. But on the table, we're looking at the RNA. So the mRNA sequence would be methionine, proline, valine, glutamine, triazine, proline. Okay. All right. Please make sure that you're able to do that, and please contact me if you have any questions regarding this lecture. Thank you.